My name is Matthew Miller. I'm the director of the Degroote Institute for Infectious Disease Research here at McMaster University, and I'm also a Canada Research Chair in Viral Pandemics. Prior to the COVID-19 pandemic, my background was really in understanding dynamics of flu pandemics. In particular, we did a lot of work trying to develop more broadly protective influenza vaccines. And the reason for that is because, of course, our seasonal vaccines don't protect us when pandemics emerge. And even more, we have to continually update our seasonal vaccines in order for them to mediate protection even on an annual basis. So we wanted to break that trend because, of course, the lack of immunity in the population against pandemic viruses leaves us really vulnerable. So a lot of that work that we'd done over the past sort of 10 to 15 years or so, we immediately sort of transferred our focus to COVID-19 when that pandemic emerged. Inhalable vaccines are really unique in many ways. There's currently only one inhalable vaccine that's approved for use right now in China. But the principle is actually pretty straightforward, right? Our propensity to give vaccines by injection is really sort of something that harkens all the way back to the days of smallpox, when we were inoculating people by scarification, by basically scratching smallpox material or scabs from infected individuals into the arms of healthy individuals in order to generate an immune response. But of course, for pathogens that affect our respiratory tract, it doesn't make a lot of sense to try and generate, this systemic immune response that's spread throughout the body. What we'd really like to do is focus the immune response at the lungs, which is the site where infection actually occurs, right? Conventionally, there is a respiratory vaccine that has been in use for quite a while now for influenza called FluMist, and it's administered as a nasal spray. And it's what we call a live attenuated vaccine. So basically, it's a strain, or it's a series of strains of influenza that have been genetically modified so that they can only infect in the upper respiratory tract and can't cause really the conventional symptoms of influenza. The problem with that approach is that viruses like influenza and COVID-19 make us sick primarily when they get into our lungs, not really when they're just in our nose and throat. And when you administer these nasal sprays, what happens is that those nasal sprays deposit vaccine only in the upper airways, so basically only in this sort of nasopharynx. And that really limits the type of immune response that you're able to generate. And so by sort of changing that strategy to deliver the vaccine as an aerosol through the mouth, we're able to deliver the vaccine through the entire respiratory tract, including deep into the lungs, where it's most important to provide protection. So far, our COVID-19 vaccine has been tested in small animal models, and it's capable of providing protection against all COVID-19 variants of concern up to and including Omicron, right? And that's very different from what we've seen with first generation vaccines that only have the spike protein of the ancestral or Wuhan-like strain of SARS-CoV-2 that essentially started the pandemic. And so these inhaled vaccines have considerable benefits in that, for one, they target a broader range of viruses, and then secondly, they also, stimulate that immunity directly in the lungs where it's most important. So currently our vaccine is in phase 1 clinical trials in humans. And what we're doing right now is what's called dose finding. We're essentially taking groups of individuals and increasing the doses so that we can figure out what the right dose is to generate what we feel is a strong and protective immune response. developing the COVID-19 inhaled vaccine and testing it in animal models prior to its approval for human trials, we did develop it as a primary vaccination. So it was sort of a one-dose mucosal vaccination of otherwise immunologically naive mice. And the reason for that is because, right, that's sort of the hardest thing to ask the vaccine to do, right? To work really well with one dose when an individual or an animal has no pre-existing immunity. And we saw that the vaccine was excellent, as I said, in protecting not only against the ancestral strain of COVID-19, but against all variants up to Omicron. However, in the current human population, we understood that almost everyone is going to either have received multiple prior vaccines and or infections by the time an inhaled vaccine is approved for use. And so as we moved into human clinical trials, we adjusted because we understood that this would be the reality in the human population, right? It would be difficult to imagine even being able to successfully recruit immunologically naive people to COVID-19 right now. What's, I think, really interesting about that approach is that there's been a lot of talk during the pandemic about hybrid immunity, right? And how good hybrid immunity can be. And hybrid immunity refers conventionally to individuals who've received vaccination and then infection, or infection and then vaccination for that matter. What happens during infection, though, to make hybrid immunity very strong is very similar to what our vaccine does, right? The unique thing about the infection is that it targets the immune response into the lungs, right? Because that's where infection has happened. And secondly, you target a broader array of viral antigens, because whereas our mRNA vaccines only target spike, right, which is just one viral protein and it's very prone to mutations, when you get infected naturally, you naturally make an immune response to all of these other viral proteins too. So it really diversifies that immune response. When we talk about monoclonal antibodies, vaccinologists would usually call that passive immunization, right? The idea is that we're giving the body antibodies passively instead of stimulating them with an antigen and having the body actively mount its own antibody response. Passive immunization is a really important piece of our arsenal to protect against infectious disease for a few reasons. One is that once you get that passive transfer of antibody, you are immediately protected, right? Whereas in the context of a vaccine, it takes time for your body to mount an effective immune response, right? Usually a couple of weeks. Furthermore, in many cases, right, you could imagine that if a new pathogen were to emerge, it might take a lot of time for a new vaccine to be approved. But if we can have broadly protective antibodies available, those could be essentially delivered almost immediately, especially to high-risk groups. It's very difficult to administer these antibodies to effectively treat these infections. And the reason for that is because with pathogens like SARS-CoV-2 and influenza virus, by the time people get really sick, it's not really the virus that's making them sick anymore. It's their immune response to the virus. And that's why in the context of the COVID-19 pandemic, for example, dexamethasone was one of the first demonstrated really effective treatments that showed clinical efficacy, right? And that's because dexamethasone is sort of a very broad-acting steroid that just dampens the immune response. The window to effectively intervene with what we would call direct-acting antivirals, meaning drugs or antibodies that work by trying to reduce the amount of virus in your system, is very narrow. And in fact, normally it's only effective before you get really ill. However, these antibodies can actually be given to high-risk people before they're exposed to prevent them from getting ill. And in that delivery model, they're extraordinarily effective. So really the optimal use of monoclonal antibodies is in settings like communal living, long-term care facilities, right? Where you have a lot of high-risk people living together in, sort of dense living conditions. Because what you could do is provide all of those people with passive immunity and avoid them from getting sick to begin with. There are limitations still, though, right? One of those limitations is that the fact that you have to deliver those antibodies by IV is not very convenient, right? It means that you have to have, trained medical professionals in there to administer the IV. Elderly people and frail people often don't have great veins. And so one thing that our group's been working a lot on as well is thinking of different delivery methods that would be more convenient, including aerosolized delivery of the antibodies, right? So in the same way that we can deliver the vaccine and target it directly to the mucosal surfaces, we can also do the same thing for these monoclonal antibodies. Find a lab, right? Reach out and get started early. I think research in this area is really exciting right now, right? Especially in the wake of a pandemic. There's never been more sort of interest in resources, so it's a great time to get into research. And I think the important thing to recognize is that you can make a real profound difference in research at all levels. In absolute terms, a very small proportion of research that ends up actually, right, in clinical trials. But our ability to have moved this vaccine into clinical trials was built on like 4 decades of research at McMaster, much of which the people doing it never would have anticipated would result in this impact, right? And so I think the other key message is you can't always predict what the impact of your research is going to be. But there's no less value working at like the early discovery phases of a project than there is working at the late phases, because all of these things build on each other.